Chapter 10, Segment 2, pages 308 through 317. Brightness is created by the visual system. Lateral inhibition. When the neurons in a region are interconnected, either through their own axons or by interneurons, and each individual neuron inhibits its neighbor. Figure 10.7b. Despite two tiles differing in color, they reflect the same amount of light. In this figure, the two tiles are not immediately next to each other. They are separated by shading. This indicates that the effect is produced by a higher in the visual system. The way we perceive and experience the world visually is created by the brain and in response to various factors, such as surrounding stimuli. Neural signals travel from the retina to several brain regions. Each eye's ganglion cells produce action potentials that travel along axons to send visual information to the brain. These axons make up the optic nerve, also known as the cranial nerve 2. The optic nerve brings visual information to the brain on each side and will eventually reach the visual cortex in the occipital lobe. All vertebrates have at least some, if not all, axons of each optic nerve that cross to the opposite cerebral hemisphere. Optic nerve cross at the midline of the optic chiasm. The optic chiasm was named after the Greek letter chi because of its crossover shape. In humans, axons from half of the retina toward the nose, the nasal hemiretina, cross over to the opposite side of the brain. The other half of the retina, toward the side of the head, the temporal hemiretina, projects its axons to its own side of the head. The distribution of these retinal fibers means the right side of the brain receives information from the left half of the visual field, and the left side of the brain receives information from the right visual field. Our eyes covering overlapping sections helps us judge the distance of objects by comparing information from the two eyes. Rabbits have a broader visual field because their eyes are farther apart. The broader visual field helps to detect predators. Most of the optic nerve fibers cross at the midline. Axons pass optic chiasm and come to be known altogether as the optic tract. Most axons of the optic tract in mammals terminate in cells in lateral geniculate nucleus, also known as the LGN, which is the visual part of thalamus. The LGN's neurons, axons, form the optic radiations which terminate in the primary visual cortex, V1, of the occipital lobe. V1 is sometimes known as the striate cortex because of a broad stripe or striation is visible, and it represents layers 4 of the cortex, which is where the optic radiation fibers arrive. Information from the two eyes come together on cells beyond layer 4 of V1, and this makes binocular three-dimensional vision possible. Some retinal ganglion cells send their axons to superior colliculus in the midbrain, which help coordinate rapid movements of the eyes toward a target and control the size of the pupil. The extrastriate cortex is the visual cortical areas outside of the striate cortex. These work in parallel to process different aspects of visual perception, such as form, color, location, and movement. In the striate cortex, as well as many other areas of the extrastriate cortex, the projection of the retina is topographic. The retina projects the brain in a topographic fashion. As visual information is carried to the brain, the point-to-point correspondence between neighboring parts of the visual space are maintained and form a map-like projection. Topographic projection of visual space due to the foveal region. Humans' V1 is located mainly on the medial surface of the cortex, and about half of it is devoted to the fovea and the retinal region around it. This representation is why we have great acuity of spatial discrimination in the central part of the visual field. We see better in this region because we devote more brain circuits to analyze it. The orderly mapping of the visual field, retinotopic mapping, at various levels means that damages to part of the visual system can be diagnosed by defects in that perception of vision. So by knowing where the damage occurred, it can be predicted where the location of the perceptual gap or the scotoma will be. A scotoma is not perceived as darkness, despite the word meaning darkness in Greek. It is a spot where nothing can be perceived. Usually rigorous testing is required to demonstrate its existence. Blind sight. Visual cues cannot be consciously perceived, but limited visual discrimination in this particular area is possible. In some cases, stimuli that cannot be seen within a scotoma and affect judgment of stimulus outside of it. In other cases, blind sight can affect the result of hemispatial neglect of an opposite side to an injured cerebral cortex. Neurons at different levels of the visual system have very different receptive fields. The receptive field consists of a stimulus region and the features that excite or inhibit the cell. The nature of the cell's receptive fields can be a good indicator of its function and perception. Photoreceptors excite some retinal neurons and inhibit others. Rods and cones both release the synaptic transmitter glutamate at their resting potentials. Light hyperpolarizes photoreceptors and they release less glutamate. The response of the bipolar cell that receives this glutamate differs based upon the type of glutamate receptor it has. You see, glutamate depolarizes one group, but hyperpolarizes another group. These groups differ in the receptive fields. Groups of bipolar cells. There are two groups, the on-center bipolar cells, which by turning on a light in the middle of its receptive field will excite the cell because it receives less glutamate, which in turn inhibits the on-center bipolar cells. Off-center bipolar cells are the opposite. By turning off a light in the middle of its receptive field, it will be excited because it receives more glutamate, and this will depolarize the off-center bipolar cells. Bipolar cells release glutamate, and this always depolarizes ganglion cells. On-center ganglion cells is when light is turned on, the bipolar cells hyperpolarize these cells. And off-center ganglion cells is when the light is turned off. The bipolar cells depolarize these cells. The stimulated ganglion cells will then fire nerve impulses and report light or dark to higher visual cortex areas. Neurons in the retina and LGN have concentric receptive fields. The pattern of connections of photoreceptors to bipolar cells influences the shape of the ganglion's receptive fields. Receptive fields of retinal ganglion cells are concentric and consist of a circular central area with a ring around it. Through many different retinal connections, photoreceptors in the middle area and those in the ring around it have the opposite effects of the cells following the circuit. Both bipolar and ganglion cells have two types of retinal receptive fields, the on-center off-surround and the off-center on-surround. The center and its surround antagonist effects explain why uniform illumination of the visual field is less effective in activating ganglion cell than a small spot that has been well placed or a line edge passing through the center of the cell's receptive field. The primate LGN has 6 main layers and some smaller layers in between. This structure is known as geniculate, because the structures are bent like a knee, which is known as genu in Latin. The 4 dorsal, which are the outer layers of the LGN, are called parvocellular because of their cells are small. The 2 ventral, which are the inner layers, are called magnocellular because the cells are large. All layers of neurons have concentric receptive fields. Most neurons in the magnocellular layers have large concentric zones and receive input from large ganglion cells, which receive their input from bipolar cells, contacting many neighboring photoreceptors. These do not show differential wavelength responses, which means they can't be involved in color discrimination. Neurons in parvocellular layers have small receptive fields, and their input is based upon cones. These neurons can discriminate in wavelengths and thus are involved with color discrimination. Neurons in the visual cortex have varied and complicated receptive fields. Neurons from LGN send axons to cells in V1, but the spots of light that are effective stimuli for ganglion cells of the LGN are not for cortical cells. 1959, David Hubel and Torsten Wiesel report that visual cortical cells require more specific stimuli that are more elongated than those that activate the LGN cells. Most cells in V1 respond best to lines and bars in particular positions and orientations within the visual field. Some cortical cells also require movement of the stimulus to respond. Some cells require any motion, whereas other cells require motion in a specific direction to respond. Cortical cells can be categorized by the stimulus that produces the greatest response. Simple cortical cells are also known as edge detectors or bar detectors because they respond best to an edge or a bar of a particular width and a particular orientation in a particular location, whereas complex cortical cells have elongated receptive fields and respond to a bar of a particular size and orientation anywhere within a larger area. The categorization is hierarchical. More complex events are built up from inputs of smaller ones. For example, a simple cortical cell receives input from a row of LGN cells, and complex cortical cells receive input from a row of simple cortical cells. Some theorists build on this idea and suggest that higher-order circuits of cells could detect any possible form by integration of enough successive levels of analysis. A neuron can be constructed for facial recognition. An hierarchical system like this, though, would require many cells, maybe even more than the cortex possesses, to account for all visual objects that we may come across.